Welcome from the U.S. Fish and Wildlife Service's National Conservation Training Center here in Shepherdstown, West Virginia. My name is John Osana, and I'd like to welcome you to our webinar series held in partnership with the U.S. Geological Survey's National Climate Adaptation Science Center. And today's webinar is titled Well-Traveled Bird Seeks Fun-Loving Plant, Waterfowl Seeking Submerged Aquatic Vegetation. And we're excited to have Kristen DeMarco, who's with Louisiana State University, with us today. Please join me in welcoming Sean Carter, who is a senior scientist with the National Climate Adaptation Science Center, who will be introducing our speaker today. Sean?
Great, thanks, John. And welcome, everyone. It's our pleasure to have Kristen DeMarco give a presentation today. Kristen's a coastal ecologist that studies the effects of environmental conditions on aquatic communities. She just defended her PhD dissertation from LSU, so in relation to spatial temporal processes that drive submerged aquatic vegetation distribution across the northern Gulf of Mexico. Previously, Kristen's worked as a coastal resource scientist with Louisiana Coastal Protection and Restoration Authority, where she assisted with the development of the 2012 Louisiana Coastal Master Plan and helped create technical recommendations for incorporating sea level rise estimates into restoration project planning and design. Kristen has a master's in environmental management from Duke and also a bachelor's from University of North Carolina at Asheville. So without further ado, please join me in welcoming Kristen today. Thank you.
Thanks for that, Sean. Hey, everybody. Thanks for calling in. So I'm going to talk a little bit today about waterfowl habitat and how submerged aquatic vegetation feeds into that in the northern Gulf of Mexico, and get into some potential effects of climate change on those habitats. The title, you guys will have to excuse me, it was kind of kitschy and cute. I just couldn't help myself, and I thought it was a little, I was a little unclear on the audience when I began this presentation, which you guys may you guys may see as I go through, but just bear with me. So, all right. So, waterfowl are, obviously very connected to water, and they use a lot of different types of wetlands in their annual cycle. The figure that you guys can see is from USGS, and it catalogs the wetland types across the United States. Everything that you see is in green is going to be a wetland. Everything in blue is going to be a little bit more deep water. And then you see some striped patterns, and that's going to be areas that have a high density of smaller, more isolated wetlands. And so waterfowl throughout their annual cycle will use a lot of these different habitats as they migrate. They breed up north, a lot of them in the prairie pothole region of the northern United States and Canada. And they breed up there and they molt up there. And then they'll do their fall migration and they'll cruise down the Atlantic coast, and they go and, get some resources from the coastal wetlands down there. They end up a little bit further down south where I am in the coastal wetlands of the northern Gulf of Mexico, and this is where they spend a lot of time. They're not breeding, they're molting, and many of them, most of them in fact, are undergoing pair formation. And then they do this whole thing again where they cruise back up north and, either through the central, Mississippi alluvial valley, or they go up on the west coast, and then they end up back up north, and the whole process starts over again. So across these different habitats, these different wetland habitats, they're using different food resources at each of them, depending on what they need. So when they're up in the north, they need a lot of protein. So they're eating a lot of really, really, fast-moving lipids. And then as they move back down and they start their fall migration and they get into their wintering habitat, they're more interested in carbohydrates. And so what I'm going to focus on today is this northern Gulf of Mexico area where the birds are spending most of their time, and they're spending it in the winter, and they're eating a lot of carbohydrates in this area. I need to get a pointer going. Can anybody see that? Yeah, there we go. So this is kind of where I am. This red box is the whole area that we're interested in, or that I'm going to be referring to today. So the wintering habitat in the northern Gulf of Mexico is home to 95% of the United States gadwall, mottled duck, 80% of green-winged teal, 80% of the redheads come through here. It's a really important migration habitat for blue-winged teal, and it supports a ton of nectin, different sorts of fish and invertebrate species. And this is really, really important. This habitat and the quality of this habitat is really important for waterfowl in the winter because the winter is really energetically expensive. It's cold, so their metabolism has to work a little bit harder. They're undergoing pair formation, which especially for the male ducks is quite exhausting. They just kind of have to follow the female around and hope it works out and they get food. They're molting. There's just a lot of expensive stuff that's happening. So having high quality habitat and a lot of food in this area is really critical to ensure the survival so that these bird species will actually make it so that they can eventually get to breeding. So what makes this high quality habitat? What makes good wintering habitat in the northern Gulf of Mexico? Well, there are these three things that they have to have. They have to have water, they have to have cover, and they have to have food. And so we have a couple of different options for, water body, depending on the species of bird that you are. If you're a diver, if you're a dabbler, you might be in a bay, you might be in a marsh, you might be in a lake. For cover, there are going to be a lot of dabbling species in particular, like this idea of a Hemi Marsh, where you have 1-to-1 ratio of open water to wetland vegetation. You can see here we've got some vegetation, but we also have a lot of open water. And it looks pretty shallow, and in a lot of cases down here in particular it is. And in these aquatic habitats is where, in the northern Gulf of Mexico, is where these waterfowl species are getting a lot of their food. So there are a lot of insects that are hanging out on the plants. The dabblers are diving down and scooping it up, munching it off. But more so in this season, they're getting a lot of their resources from eating seeds, from eating roots and shoots, tubers, from aquatic plants. And a lot of these aquatic plants are going to be submerged aquatic vegetation. So they'll eat, emergent plants as well, but this guy right here, these submerged plants that spend the entirety of their life cycle under the surface of the water, with the exception of flowering, are going to be where a lot of that carbohydrate nutrients are going to be, they're going to get it from. And really, when anybody's trying to calculate, okay, like how much habitat do I need? When I say anybody, I'm talking mostly about waterfowl and coastal managers. When these guys are trying to figure out how much habitat they need to support a target waterfowl population, they really need some estimates on, what's going on with the SAV in this area, because the SAV is going to be really critical to this energy density component that is put into these calculations for habitat objectives. So basically you figure out how much a given bird, a single bird or a group of birds needs to survive the winter, and then you figure out how much energy you have based on your food resources, and then you can calculate how much habitat you need to support whatever population you guys are interested in. So unfortunately, there are not a ton of studies in the northern Gulf of Mexico that quantify these values. So there was a study that was done in 2003 by, he was an LSU guy, Christian Winslow, and he studied fresh marsh, and he found this value of 240 kilograms per hectare for food resources in aquatic habitats and fresh marsh. So this is seeds and submerged aquatic vegetation. And then what, based on other information, waterfowl managers, and I'm referring in large part to the Gulf Coast Joint Venture managers here, but it's kind of the baseline for everybody what they go from. So they had just assumed that intermediate was about equal to fresh marsh in terms of food resources. Brackish was about 50% equal to fresh marsh, and saline marsh zones were about 10% equal. And so the idea here is based on this theory that there's a giving up threshold or a giving up density where once the food density, the energy density goes below a certain threshold, the waterfowl they're gonna move. They're gonna bail, they're gonna go to a different area, they're gonna search out more food. Now, this theory is it's it's discussed, and it's currently being more discussed by some other people, but as of right now, it still holds. And that's what these guys are interested in. How much food do I have in this area, or how much food do I need to have before the ducks are going to leave and I'm not going to be able to meet my habitat or my population goals. So what we do know about SAV is that it's found in all different marsh zones across the northern Gulf of Mexico. And we refer to these in these 4 major categories: fresh marsh, intermediate marsh, brackish marsh, and saline marsh. So based on literature and studies, we know that it shows up in saline, it shows up in brackish, it shows up in intermediate, and it shows up in fresh. And we know that the distribution of SAV is affected by, it has spatial and temporal variation that is affected by salinity regimes, water levels, temperature, precipitation, geology, and various sorts of human activities. And so out of these different sorts of environmental drivers that can impact SAV distribution, a lot of them are going to be changed, altered. Variability might be affected by climate change. Salinity regimes, water level, temperature, and precipitation, the heavy hitters here. So we know that these areas are going to be affected. We know this coastal landscape is going to be affected by accelerated sea level rise. Most of the models predict, a minimum of 0.5 meter up to 1.5 meters. So we're very unsure about what the submerged aquatic vegetation response to sea level rise is going to be. What we do know is that in this area, everything that you guys can see in red on this figure is 0 to 5 feet above sea level. And this is in 2008. This is from a 2008 data set, so who knows what it looks like now. Our bathymetry data and our elevation data in Louisiana is not super, but we do know that this area is very low lying. So even if we do only get 0.5 meter of sea level rise, it's still going to change the landscape considerably. And what that could mean for these different marsh zones is that we could see a shift, landward migration of, saline habitat, fresh. Everything could essentially move up. What we could also see is we could just see land loss. Now, this is a climate change scenario projected out to 2060 from the CPRA, Louisiana Coastal Protection Restoration Authority 2012 master plan, and that's assuming, about 0.5 meter of sea level rise. And this is, everything that you see in red is basically going to be converted to open water under this scenario. And so, instead of these marsh zones moving up, they could just convert to open water, which for submerged aquatic veg, that might just be more habitat. But we just don't know at this point because, again, we don't have a lot of quantifiable data on that. So everything that's over here in pink, we identified as potential submerged aquatic vegetation habitat. Now, these are shallow, in general, under 2 meter waters. These are interior coastal ponds, and that's especially the interior coastal ponds are really important because they do support a lot of SAV, and they also support a lot of waterfowl, and it's a really important habitat for those guys as well. So again, looking at these different marsh zones, what some models are predicting, this is the sea level affecting marsh model that was ran for the entire Gulf of Mexico. They're predicting in some cases, up to a 60% decrease in fresh and tidal fresh marsh that would include intermediate and fresh marsh, and almost a 40% decrease in interior fresh marsh. So these really critical interior habitats, the intermediate and the fresh marsh, could be completely lost in the event of sea level rise, or they could migrate. But again, we need some quantifiable data to figure out how that's going to affect waterfowl. So what's the actual habitat quality of marsh zones for waterfowl? This fresh study that I mentioned earlier, that was all done in interior coastal ponds, 240 kilograms of food per hectare, but we really don't have any data on the rest of these marsh zones. So what my project did is we went across the northern Gulf of Mexico and we divided it up spatially into regions. So over here, this is the Texas Mid-Coast region, and these are all based on Gulf Coast Joint Venture Initiative areas, which are based on waterfowl and ecological data. And anyway, this is the Texas Mid-Coast. This is the Chenier Plain, all this area over here. This is the Mississippi River coastal wetlands, and then this is coastal Mississippi and Alabama. And I'll refer to these regions a few times coming up. We randomly generated individual sites across the coast, and what we did was we sampled about 384 sites during the growing season every summer for 3 years, and we collected a whole bunch of data. We got species data, presence data, percent cover, depth, salinity, turbidity, temperature. And we compared SAV presence and cover and by region, by marsh zone, and by year to try to figure out if there were some patterns that we could pull out. And just very generally what we found, mean percent cover was 16% coastwide for all 3 years. And over this time period, all of these, any differences that we found, any significant differences were all spatial. There was no temporal difference in SAV presence or cover. Now, if you guys remember 2013 through 2015, there were no big storms either. So certainly if there had been a storm, we probably would have had a temporal change. And if the scale had been a little different, or if we'd been looking at one species, right, instead of like an assemblage, it might have been different. Okay, so over here, the figure on the right, this is percent SAV presence. The figure on the lower left is percent SAV cover. So cover, presence. I'm going to talk about presence first. There was a significant interaction between marsh zone and region. You see that we have some of these regions that have much higher presence than the other regions, namely the Mississippi River coastal wetlands had about equal presence in fresh, intermediate, and brackish marsh. The Texas Mid-Coast region in the dark had very, very high presence in fresh, and then much lowered presence in intermediate, brackish, and saline. And then what we also have is we have a pretty significant difference in cover between fresh and saline. So fresh, we had higher cover, saline, we had lower cover, but you can tell that, you know, we look at the Texas Mid-Coast and the coastal Mississippi, Alabama, these areas had much higher cover in the saline marsh types than Mississippi River or the Chenier Plain, right? So we see some very significant regional differences that are also affected by marsh zone. Okay, so this, you just kind of have to trust me a little bit. These are species codes. These are taxonomic species codes. It moves from fresh up to saline, right? So the fresher species are down here, the saline are down here. This is the Texas Mid-Coast. This is the Chenier Plain. This is the Mississippi River, coastal Mississippi, Alabama. And then down here we have fresh, intermediate, brackish, and saline. So this is just kind of a qualitative analysis. I want to describe some of the marsh types to you guys. What we have over here, this is all of our fresh marsh. Everything that you see in blue, fresh. You notice right away that a lot of it is concentrated in the Chenier Plain and in the Mississippi River coastal wetlands area. And what we have in this fresh marsh is we have a lot of different species, and we also have a lot of hydrilla. That's this guy right there, hydrilla. And we have a lot of coontail, Ceratophyllum demersum. That's that guy right there and that guy right there. And really, really in a high percent cover in the Mississippi River coastal wetlands and in the Chenier Plain. And we see that we don't have that over here in the Texas Mid-Coast, and we don't have that in coastal Mississippi, Alabama. In fact, in coastal Mississippi, Alabama, we mostly just have Rupia maritima, or wigeon grass, which shows up. So what we start to see is that there are some differences in species assemblages across regions. One of the environmental variables that we did pull out in fresh marsh was that fresh marsh was significantly deeper than all of the other marsh types, and significantly deeper in the Mississippi River coastal wetlands area than the other areas. So fresh was always deeper, and in the Mississippi River it was real deep. So we lumped intermediate and brackish together. This is all of the intermediate. Again, you see a lot of intermediate marsh in the Chenier Plain. And then these are the intermediate species assemblages. We see an increase in species assemblages for the most part, with the exception of coastal Mississippi, Alabama. And then this is the brackish. And one of the things I want to point out is you'll notice Texas Midcoast, there was almost nothing to pick from in terms of what was defined as being brackish marsh by the data set that we used, which we used NRIGHT 2014, if you all are familiar with that. And if you want to talk about it more, I can tell you. But anyway, the species types that we had in this intermediate and brackish, we had a lot of rupia, a lot of widgeon grass. We had a lot of milfoil, it's Eurasian milfoil, or Myriophyllum spicatum, and then we had a lot of Nahas guadalupensis. And so what we noticed is that even, we would have these similar numbers of species assemblages. The habitats looked pretty different, right? So all of these habitats supported Rupia maritima. This is Texas Midcoast. This is Chenier Plain. This is Louisiana, and this is Alabama. So this is, really, really deep pond with Rupia that was probably about 2 meters. This was a little bit more stubby like lawn. You see some species will kind of show up everywhere. And that's kind of the point there. So moving on to saline marsh, you can't see it very well because, again, it's saline. It's all kind of on the fringe. And what we immediately notice is that saline marsh, we drop down in the number of species that we have. However, these are, we have some seagrasses that show up. Haliduli rhydi, or shoalgrass, shows up in the Texas Midcoast and in Alabama. We have some interesting, we have turtle grass show up in coastal Mississippi, Alabama. So even though the species assemblage goes down, we don't see any of those invasives, that we saw in some of the other areas. And saline marsh looked really different depending on where you were. So saline marsh, Texas Midcoast, Chenier Plain, Louisiana, and then in Alabama. So one of the other things that we needed to look at was seed biomass, because we're talking about, we want to know what the birds are, what they're able to eat. And we also found that there was no temporal effect at the scale, and we also found a significant difference by region and marsh zone. So what you can see here is that Mississippi River coastal wetlands, really high seed biomass in the intermediate, Texas Midcoast, really high and fresh, and then everything kind of starts to go down from there. So you have these kind of hot spots depending on where you are. And then this is all food. So this is seeds that are considered to be waterfowl food. And these are just submerged aquatic vegetation seeds, right? So not all of the food. So if you look over here at Texas Midcoast, this is, 2.5 grams per meter squared. The SAV seed is a little bit under 1 gram per meter squared. So not all of this food was necessarily SAV seed. What we had in the fresh zone of the Chenier Plain, we had a lot of Cyperaceae seeds. In the fresh zone of Texas Midcoast, we had a lot of Chenoplectus seeds and a lot of Nymphaea species seeds, so a lot of lilies, things like that. In the fresh zone of the Texas Midcoast, we had a lot of Stachenia pectinata, which is that sago pondweed, really, really good wildlife food. Potamogeton species in Mississippi River, Rupia maritima, contributing really almost entirely to this big food biomass in the Chenier Plain area. Some of the other cool things that we looked at was that increasing submerged aquatic vegetation cover was correlated with increasing food seed biomass. And that's not just SAV seeds, right? So we know that, you're in an SAV bed, and so you're getting, all of these SAV seeds are being deposited on site. There's some little Rupia seeds right there if you can't see them. But what we're also seeing is that the SAV beds are trapping other seeds, and not just anything, well, it's trapping a lot of different things, but it's trapping a lot of food. So there's a lot of stuff, if you're a bird, there's a lot of stuff to eat if you're in an SAV bed. So based on this literature, we knew that there are, there's some key drivers for SAV, well, based on the study and literature. So what we see is, we primarily saw a big effect of salinity, based on marsh zone. We know that light's a big deal from both our research because of the, some depth stuff that we found, and then, other people have found, light is just very, very important for submerged plants. And then exposure, also seemed to be pretty important. So depending on, what was going on with waves, what was going on with wind-driven waves, where you're going to find SAV is sort of relevant to that. And what we wanted to do was we wanted to take all of these individual sites and we wanted to say, okay, well, can we take this information that we learned and predict SAV distribution across unsampled areas? And that's what you really need to know if you're trying to predict how these habitats are going to be affected by climate change, because understanding these drivers of presence can tell us a lot about SAV and a lot about waterfowl habitat and how it might change in response to changing climate. So enter the species distribution model. So species distribution model takes occurrence data, so observational data, basically what we collected in the study I just detailed, and it relates it to environmental characteristics. And these are also called niche models, so that's why you'll see the word niche here a lot. Basically, you have one condition, say that's salinity, and then you have another condition, and you say that that's exposure, and you find all of your presence data, and you basically are able to quantify, the likelihood, the probability that that species or that assemblage of species that share similar niche requirements, that have similar environmental requirements, you basically figure out the probability that it will live there. So we did this for a distribution of SAV assemblages, so all SAV species together. And we only did this in Louisiana because Louisiana, we have access to this great continuously recorded hydrographic data from the Coastwide Reference Monitoring Station. So we collected all of this hydrographic data, salinity mean, salinity standard deviation, water level mean and standard deviation, and temperature to drive our species distribution model, and attached it to our observational data. So this is an example of what the salinity layer looks like. So we took all of that hydrographic data, we interpolated it across the surface of coastal Louisiana. This is just an example from winter 2013 to 2014. And using that salinity data, we calculated a seasonal mean and seasonal variability. And then we also wanted to come up with a proxy for light. So we used, again, as I mentioned earlier, Louisiana has terrible bathymetry data. So we used turbidity as a proxy for light. And this has been shown to work very well in other areas, but basically what you do is you use band 3 from your Landsat imagery, and that correlates really well with turbidity in other areas, particularly in kind of muddy bays and deltas. And so now we have a spatially contiguous layer for turbidity across the coast. And then we generated an exposure index. And the exposure index is fairly simple at this point. It's being worked on actually right now, but the exposure index was just a measure of fetch across an open water body. So if you had a really big open water body, you would have a really high exposure value. We weren't able to account for orientation or dominant wind or wind power at this point in time, but it was just a place to get started. And so using these data, we used a bunch of different environmental variables, but the different seasonal and variability and mean. And what the model pulled out, we did a binomial logistic model, and what it pulled out was that mean winter salinity, reflectance, and exposure were the most important things and the only things that you necessarily had to have to calculate submerged aquatic vegetation presence, or the probability of submerged aquatic vegetation presence across Louisiana. So high probability of presence, warmer colors, low, cooler colors. And this is kind of hard to see on your figure, but this is Lake Buff. I don't know if any of you guys are Louisianans, but that area is absolutely full of SAV, and it looks a little bit like the salinity map. You can kind of see how the likelihood of SAV goes down as you get out toward the coast. And that makes a lot of sense, both because of the salinity type, but also because of the exposure out there. There's just not a lot of plants that can grow out there. And so our model was significant, and it had a correct classification rate of 75%, so it was correct 75% of the time. And if you guys look over here, the error, the red stuff is going to be where the model was incorrect. So what we have is where the model predicted presence and it was absent in the field, or where the model predicted presence and it was, or the model predicted absence and it was present in the field. So just a couple zoom-ins. This is an area in southwest Louisiana that we had a pretty high error rate. That area is bizarre. There's a lot of crazy stuff that happens out there in terms of management, in terms of water level. Nobody really knows what's going on. And then this other area, you see a lot of error at the border between saline and brackish marsh zones. So there's something going on there as well. But basically what this told us was you can use a couple key conditions to predict SAV occurrence in coastal Louisiana. And you can do it pretty well. You can get a 75% correct classification rate. But what that sort of told us is that, all right, salinity is important, but it's not our whole story. And also, this whole thing is using, we're using seasonal salinity and these salinity changes and seasonal turbidity to predict presence in the summer season. And this is South Louisiana. So we wanted to see if there was any benefit to sampling at a finer resolution, at a finer scale. So we took a smaller area in Barataria Bay, which is just this area that's just west of the Mississippi River, and we sampled every 6 to 8 weeks in 2015. And we collected the same data that we collected in the big data set, presence, absence, turbidity, depth, temperature. They were more meaningful, though, because we collected them every 6 to 8 weeks instead of just once, during a growing season. And so we collected information on SAV and on FAV, which is floating aquatic vegetation. And we evaluated the effect of marsh zone season and the influence of FAV cover on SAV cover. And so again, what we found, this is the mean salinity over the season. This goes from early spring all the way to late winter. Salinity makes sense. The purple is saline, green is brackish, light blue is intermediate, dark blue is fresh. Salinity all makes sense. There was a significant effect of marsh zone. There was a significant effect of marsh zone and season on depth. So you see, again, fresh marsh zones were deeper. And in coastal Louisiana, we have these cold fronts that come in and they decrease the water level pretty significantly, and we were able to pick that up in our data. Temperature, we see that temperature totally makes sense, right? Total temperature across season, right? We also see cooler temperatures in the fresh marsh zone, and again, that's probably because it was deep. And then there was no effect of marsh zone or season on turbidity. Turbidity, there was nothing really interesting going on there. So like the big data set at this scale, all the drivers of SAV were spatial. There was no effect of season when you looked at all of these different marsh zones. What we saw is this is SAV presence. We saw equal presence in fresh, intermediate, and brackish marsh zones, decreased presence in saline. This is percent cover. We saw equal percent cover in fresh and intermediate, a little bit lower in brackish and saline. And this is for SAV. So looking at FAV, we found that FAV really only occurred in the fresh marsh zones. So if you look up here, we've got fresh, intermediate, brackish, and saline. And you notice you don't see any brackish or saline on here. We had floating aquatic vegetation only occurring really in the fresh marsh zones, and to a very, very small degree intermediate. And there was a significant effect of season on FAV. And so what we wanted to do is I wanted to look at just submerged aquatic vegetation in fresh marsh, and there was a significant effect of season and floating aquatic vegetation on submerged aquatic vegetation in fresh marsh. This red is SAV over time. This blue is FAV over time, and you can see that as FAV increases, SAV starts to decrease. And then as FAV starts to decrease, SAV starts to increase. And this makes sense. If you guys work in these areas, it's kind of intuitive, but it hasn't really been quantified before. And it was kind of cool just to find that it's spatially specific to this one area. And again, it makes sense. We see that these cold temperatures are probably essential for maintaining SAV in this fresh marsh habitat. This is temperature. This is season. This is in the winter. We see all the vegetation is dead. There's no floating aquatic vegetation that's alive on the surface of the water. Temperatures are low. This is the early spring. Everything's starting to green up nicely. You can't see it from this photograph, but there's SAV starting to pop up. We probably had about 30, 40% cover at this point in time. we start to get into the summer and you see this massive increase in floating aquatic vegetation. This is all hyacinth. It was about 1 meter high. And at that point, all of the SAV under the surface of the water is completely dead. So increasing invasive FAV cover significantly decreases SAV cover. And it makes sense, but there was a clear temperature pattern that was associated with it. So the main point that I want to make is that habitats supporting SAV are affected by multiple effects of climate change. Sea level rise, for sure. So these low-lying areas, these are going to change the distribution of open water, marsh zones, things like that. It's, with these changing flood risks, that's going to change exposure values and how much the individual plant is affected by waves and winds and things like that. We're also dealing with temperature increases, and temperature increases may also affect species assemblages. Precipitation changes will absolutely affect species assemblages. And then we also have, us running around down here putting in marsh restoration projects, putting in terraces, putting in freshwater diversions, putting in sediment diversions, and all of these efforts are going to have an impact on the habitat and on the environmental variables that determine whether or not you have SAV or you don't, which in turn can create differing types of waterfowl habitat. So waterfowl are great. They're super mobile. They can move to adapt. These are two examples of sites that we visited. This is Grand Bay, Mississippi. This is San Antonio Bay, Texas. So, if a project goes up or an area floods or it's not, optimal habitat, the bird can move, right? And it will do that, but habitat configuration is also really important to waterfowl. So you need certain habitats to be within a certain distance of one another. Particularly, the bird that springs to mind is redheads. Redheads have to have fresh water, or they need fresh water within 8 kilometers of seagrass beds. And so this is an example of Mississippi. And what's kind of happened here is we have this road here that's really hydrologically cut off this area, so you don't get this nice transition into fresh marsh anymore. You just have seagrass, nothing, and then you get some fresh stuff up in here. And in Texas, they're undergoing this big drought right now, so they have these beautiful seagrass beds in here, and then virtually no fresh water to sort of deal with. So, natural and anthropogenic changes, they can decrease habitat quality, especially when you think of it in a sort of a larger scale. And the main point that I want to get across from this seminar is that there are multiple characteristics that determine habitat quality. It's not just salinity regime. It's landscape configuration, it's species assemblages, it's the combined effects of these environmental variables in estuaries, the proximity to humans. And basically the takeaway from that is that while fresh marsh may support the highest SAV cover and seed biomass, these areas might not always be the highest quality habitat, depending on where you are. So if you remember the Mississippi River coastal wetlands area, we had these really deep ponds that were just absolutely loaded with hydrilla. And then you'd go over to the Texas Marine Coast and you'd have these shallower, somewhat shallower fresh ponds that were full of Stachynia pectinata, which is a much more desirable waterfowl species. And then we also have to consider these other macroclimatic drivers affecting these aquatic habitats. So these increasing temperatures and increasing sea level rise, as they impact fresh marsh, they may significantly increase the importance of intermediate and brackish habitat, especially if we see this increase in invasive floating aquatic vegetation species limiting submerged aquatic vegetation's ability to thrive. Okay, that's going to do it for me. This project was funded by the Department of Interior and the USGS South Central Climate Center with support from the Gulf Coast Joint Ventures and the various LLCs that you see listed on here. I'm still hosted at the LSU Ag Center, and so I always have to thank them for putting up with me. And I appreciate you all for calling in on this hot summer day. And that's all I got, and I'll take questions.
Thank you, Kristen, and as she mentioned, she will be taking questions, so if you have any, feel free to throw them into the chat box. So, first question is from Lindsay VZ. What was the most surprising finding of this research?
I think it was the difference between fresh marsh, depending on where you were, because like we've always been under the assumption, or at least I was, as a bird person and as a wetland person, that these fresh marsh habitats are so high quality and they're going to be the most desirable thing. And really in a lot of areas, it's not, you go in there and you're like, this is not anywhere a duck is going to want to be. This is not anywhere I want to be. But it's just these super isolated ponds that are incredibly incredibly deep, and they're just loaded with a lot of invasive species. But then you go to a fresh marsh zone in a different area, and it's perfect. And a lot of that is regulated by salinity, but again, there are all these like multiple factors. The depth is really important. But that, I think, was the thing, is that fresh marsh in the Mississippi River coastal wetlands versus fresh marsh in the Texas mid-coast, they're not, in my mind, they're not really comparable in terms of anything, really. They're totally different. So I mean, and we're always, as ecologists, we're always struggling to try to classify things, especially spatially, to make things easier on ourselves. But that was the thing that really, stuck out for me, is that they're super different, and they should be treated as such.
Thank you. And we have a question from Norman Johns. Did your species sample include Vallisneria americana, only one population known in Texas?
Yes. There was, you're right, John, there was not a lot of Vallisneria in Texas. Most of the Vallisneria was in the intermediate zones of the Mississippi River. I know you told me how to do this, John, but I didn't write it down. Okay. Most of the Vallisneria was in the intermediate zone of...
Kristen, you can click on that box next to the right-hand arrow.
This guy? Oh, the draw? That's what it was. Okay. Well, I'm where I want to be anyway, so... Okay, can you guys see it?
The arrow?
There we go.
You can see the arrow.
Okay. So this is Vallisneria. This is Texas mid-coast. So we had a little hit of Vallisneria over there. This is the Chenier Plain, a little hit of Vallisneria over there. This is the Mississippi River coastal wetlands, and we had Vallisneria across fresh intermediate and brackish marsh zones over here. So we were able to pick some up in Texas. I'm trying to remember where on earth that was. I could probably find out. But yeah, we were able to find it, but it was very sparse in Texas, to say the least.
Okay. Thank you. I saw someone typing, so we'll see if they want to follow through with that. While we're waiting to see if anyone else has any more questions, I would like to say thank you again to Kristen. I'd like to thank our partners at USGS and all of you who participated. Thank you for attending today. And let's see, doesn't look like we have any other questions coming through the chat box. So on that note,
there we go.
We go.
Hey, Ken.
There we go. I saw somebody typing. So nice talk, Kristen. How does the increase in black mangrove factor into food availability in brackish salt marshes?
Yeah, well, that is going to be kind of the next hot topic, right? Is what do we do about mangroves? Particularly here in Louisiana, everybody's really excited about mangroves because they provide that additional shoreline protection. But again, mangroves in these brackish habitats, it's going to shade out any possibility for SAV. Right now, it's kind of a non-issue because the SAV is so patchy in our brackish habitats. We don't get these nice meadows like we would get in saline marsh types. And so having, I think having some mangroves show up, they actually might provide a little bit of that, that landscape that would create more protected sort of coves where SAV could show up. But I don't. But I honestly don't know what's going to happen with that now. We had a bunch of mangroves in Louisiana, and they all got killed this winter, so they were all murdered. And no, ducks do not consume Avicennia propagules. It would be nice if they did, right? Because they're these huge, big, fat seeds. But no, they don't eat them.
Okay, that sounds like a good question to end on. Let's see, Norman typing away. So I just want to see real quick if that's a question. Just a thank you. Excellent. Thank you both. Thank you everyone for your questions. And thank you, Kristen, for the presentation.